Here we'll learn about the vascular system, which transports blood throughout the body. To begin, start at table. To note that the vascular system comprises the following components: the arterial system, which includes arteries and arterioles; the capillary system, which comprises the capillary beds, which are complex networks of tiny vessels; and the venous system, which includes post-capillary venules, and veins. These vessels join to form a closed system through which the heart pumps blood. To note that the lymphatic system comprises a series of lymph vessels and nodes that works with the circulatory system by draining excess fluids and proteins from the tissues and returning it to the blood. We'll briefly mention the lymphatic system in our diagram, and you can learn more about it in our notes. Let's illustrate the general organization of these vessels within the vascular system. We'll start with the arterial side, which carries blood away from the heart. Show that the largest arteries, which includes the aorta and its large branches, are called elastic arteries due to their size and relatively low resistance. They're considered conducting arteries. The elastic arteries branch and give rise to muscular arteries, which deliver blood to specific organs. They are distribution arteries. Muscular arteries branch and give rise to small arteries, which then give rise to arterioles, the smallest of the arteries. Neural and hormonal stimuli alter the diameter of the small arteries and arterioles to alter the resistance to blood flow and therefore its delivery to the capillary beds. They are resistance vessels. The diameter of these vessels is a key factor in systemic blood pressure. Next, show that arterioles give rise to meta-arterioles, which lead to capillary beds. Show that precapillary sphincters regulate and direct the flow of blood from the meta-arteriole through the capillary beds. Capillaries are the smallest of the blood vessels and facilitate molecular exchange between the tissues and the circulation. Capillary bed blood flow alters to meet tissue demands. Now, at the distal end of the meta-arteriole, indicate the thoroughfare channel. Show that the capillary bed drains into a post-capillary venule, which is the smallest component of the venous system. These vessels do not have smooth muscle or sphincters to regulate blood flow. Post-capillary venules merge to form larger venules, which ultimately merge again to form small and medium veins and then large veins. Indicate that veins have high capacitance, meaning that they can accommodate a large portion of the total blood volume at relatively low blood pressures. Pause and indicate that anastomoses, which are direct connections between the small vessels, can form. We show an arteriovenous anastomosis between small arteries and veins, which are abundant in the skin and allow for cooling. Finally, indicate that lymphatic capillaries take up fluid from the interstitial tissues around the capillary beds. This fluid passes through increasingly larger lymphatic vessels and associated lymph nodes before draining into the venous system near the heart. To note that blood vessel walls are generally organized into three layers: the tunica intima, the innermost layer, which contains a single layer of endothelium surrounded by connective tissue; the tunica media, the middle layer, which contains variable amounts of smooth muscle, elastic connective tissue, and collagen, which limits the distensibility of the vessel, its ability to swell; and the tunica adventitia, the outermost layer, which comprises mainly collagen. To note that elastic lamina supports the tunica adventitia, anchoring blood vessels to nearby organs and providing stability. As we'll see, the thickness and relative compositions of these layers vary between vessels according to their size and whether they're on the arterial or venous side. Now let's illustrate three types of arteries: elastic arteries, which are the largest; muscular arteries, which branch from elastic arteries; and arterioles, which are the smallest and branch from muscular arteries. Draw the cross-section of an elastic artery from inner to outer: a thin tunica intima, a thick tunica media, and the tunica adventitia. Show that the tunica media in elastic arteries contains a notably large amount of elastic connective tissue, which enables them to expand and recoil as the heart contracts and ejects blood into circulation. The aorta is an example of an elastic artery. Draw the cross-section of a muscular artery as follows: a thin tunica intima, a thick tunica media, and the tunica adventitia. Show that the tunica adventitia, the elastic and muscular arteries, has its own blood supply called the vasa vasorum. As we'll see, the outer wall of large veins also has these tiny blood vessels. The adventitia of these larger vessels need their own blood supply because they're so far from the luminal blood, which otherwise nourishes the vessel tunics. Draw the cross-section of an arteriole as follows: a thin tunica intima, a thick tunica media, and a thin tunica adventitia. Draw the cross-section of a second smaller arteriole as follows: a thin tunica intima and tunica media, which comprises a single layer of smooth muscle. Notice that this breaks the rule of 3 tunics. The smallest arterioles and venules comprise only 2 layers. Indicate that the tunica media of muscular arteries and arterioles contains abundant smooth muscle, which allows them to regulate blood flow by vasoconstriction, a contraction of smooth muscle that narrows the lumen, and vasodilation, a relaxation of smooth muscle that widens the lumen. Now let's show the vessels of the venous system, which returns blood to the heart. In general, the vessels of the venous system have thinner walls and larger lumens than the arterial system, with less defined elastic and muscular features. Indicate that we'll illustrate a large vein, such as the vena cava, which returns blood to the heart, small and medium veins, such as which we'd find in the arms and legs, and a small venule, such as which we'll find draining a capillary bed. Begin with a cross-section of the vena cava from inner to outer: a thin tunica intima, a thin tunica media, and a thick tunica adventitia. The tunica adventitia is the largest layer in veins and contains a small amount of smooth muscle, unlike arterial walls where smooth muscle is limited to the tunica media. And as mentioned earlier, it houses a vasa vasorum, vessels of the vessel. Notice that the tunica intima and the tunica media are thinner than in the elastic and muscular arterial walls. Next, draw the tunica adventitia, media, and intima of a small or medium-sized vein. Note that the tunica media is thinner than that of its complementary artery. Show that the tunica intima of small and medium veins creates specialized valves that prevent the backflow of blood. Recall that veins are under low pressure, and that in the arms and legs, for example, they're responsible for moving blood against the force of gravity. The valves help ensure unidirectional flow. There's a clinical correlation to note that faulty venous valves cause backflow of blood. This manifests as varicose veins, often seen in the legs of elderly people. Finally, in this section, draw a post-capillary venule in cross-section. Recall that this is the smallest vessel in the venous system. Show that these vessels are so small that they also break the 3-tunic rule. They comprise an outer basement membrane and an inner lining of endothelial cells. This arrangement provides a porous wall that facilitates the movement of white blood cells in and out of circulation. Finally, let's learn about 3 types of capillaries. First, draw a continuous capillary as a solid wall of endothelial cells connected by tight junctions with a basal lamina surrounding it. This thin endothelial wall facilitates molecular exchange between the lumen of the continuous capillary and the surrounding tissue. These are the most abundant capillaries in the body. Next, draw a fenestrated capillary as a continuous wall of endothelial cells with pores, which are the fenestrations, the windows. Show that a continuous basal lamina surrounds the wall of endothelial cells. Fenestrations produce greater permeability than that which we see in continuous capillaries. Fenestrated capillaries can be found in specialized tissues that require more rapid and extensive molecular exchange, such as in the kidneys and small intestines. Lastly, draw a sinusoidal capillary with large gaps between endothelial cells. Show that a discontinuous basal lamina surrounds these endothelial cells. The large gaps between endothelial cells and incomplete basement membrane permit even greater permeability than that of fenestrated capillaries. Sinusoidal capillaries facilitate the passage of red and white blood cells and are found in specialized organs within which such movement regularly occurs, such as in the spleen and liver. This concludes our diagram.